Guide for Monday, December 1st. This is the reading guide for Monday, December 1st. You have two papers that you're going to be reading for today, and I'm going to take you through in order the way that I want you to read them. Although you are only doing a message box for one paper, you do need to read both to be able to participate in the team activities. So don't just read the one that you have a message box for, but clearly pay close attention to the one that you have a message box for and do a really good job on that to help your team out. All right, we are going to start with the paper on ivory-billed woodpeckers. This is a paper that came out of the lab that I was in at the Smithsonian, although before I was there. So Rob Fleischer was my postdoc advisor, and he's the lead author on this paper. This paper came at a pretty exciting time, a time when people were reporting sightings of ivory-billed woodpeckers in North America, largely in Louisiana. So I went to a conference in Mexico for researchers of birds, ornithologists, and there were presentations of video footage, of recordings of calls and knocking on trees, with the thought that people had recorded ivory-billed woodpeckers. None of that has been confirmed, and it's been about 8 years now that we have not been able to verify that an ivory-billed woodpecker still does exist in North America. But there's a lot of enthusiasm around that, and so that's where this paper came out. A group of people saying that they had found ivory-billed woodpeckers and wanted to sequence feathers and scat that they had found from birds in this area in Louisiana. So they wanted to have DNA sequence that they could use that would identify if it was an ivory-billed woodpecker or not. So as you start reading that, now you understand where this paper was coming from in terms of the political happenings of the day. They're going to talk about their research questions, and I want to explain DNA barcoding. I just sort of mentioned that they wanted to be able to have some DNA sequences that they knew belonged to the ivory-billed woodpecker, and in particular some sequences from some short regions that are known to be unique from other closely related species. So that's a DNA barcode. It's in vertebrates from cytochrome B, which is a mitochondrial gene. So this study aims to get a sequence like that and to verify that it is unique. So they're going to give you their methods here, and I want to point out that down here, so you're just skimming a little bit, but I do want to point out that they talk about, at least I think they mention this, so the ancient DNA lab work was conducted in separate laboratories from where the modern DNA work was going on. So I want you to think to yourself, why is that an important thing to do? We can talk about that in class. Missed a page here. Okay, you don't need to know exactly how the phylogenies were conducted, so don't worry about this section right here, okay? But what I do want to point out is, you remember we talked about AIC, Akaike Information Criterion, as being a way of selecting a model. They did use that same method. I told you it would come up again, and here it is again. You don't need to know about it, but I wanted you to see that it happened again. Okay, now they're going to talk about dating their tree. They have dating of nodes. So I want to tell you what that means. You already know that there is such a thing as a phylogram rather than a cladogram. So look at this picture. Is this a cladogram or a phylogram? I'm going to point out that it has a legend here telling you the length of a branch and how many substitutions it correlates with. So then you're going to know that this, of course, is a phylogram. That the length of these branches corresponds to evolutionary change, which we think also corresponds to time. The mutations in DNA sequence, of course, these trees are made from DNA sequences. Mutations accumulate at a rate more or less constant over time. There's a lot more to that, but I'm going to give you sort of the basic instruction on that. So what you can do once you have a tree that is a cladogram, not a phylogram, is you can look at the rate of mutation along branches, and you can then say that rate, if you know for this gene, for instance, most mitochondrial genes, particularly cytochrome B, it's been estimated to mutate at a rate of 2%. So 2% sequence change per million years. So if you take two species that are sister here, we have pileatus here and we have lineatus, the amount of difference between them, say in 100 base pairs, you have two differences. That's 2%, 2% change. Then you would assume that this divergence here, this node, this divergence happened about 1 million years ago, because we think the rate of change is 2% per million years. Okay. And that's been estimated from other studies. You don't need to really know exactly how we got that number, but that's a more or less agreed upon number, which I would love to spend hours talking to you about, since that's an area of my active research, but we won't go there for now. So how can you apply this rate across your tree? You can simply enter in that 2% time, and then you can use some software to tell you what time period these different nodes happened then, if you know that 2% is your rate. You can also implement a fossil or a geographic constraint. So say we know that these two species, pileatus and lineatus, for instance, are found on opposite sides of a mountain range. You could say that the divergence would not have happened until that mountain range had risen. Or if they were found on two islands at one time were connected, you could say that these two species did not separate until those two islands separated, right? Until one island became two islands. So you could constrain this and say that this happened 5 million years ago. So I know that this divergence is 5 million years old, and so then I can assume that this rate of change, I can recalibrate that rate of change based on knowing this time here for this node. So they did estimate divergence dates on this phylogeny. They estimated them in a number of different ways. They applied a 2% sequence divergence rate, like I explained. They also applied a constraint where they said here, isthmus formation, okay? So the Isthmus of Panama formed 3.1 million years ago, and so because we know that woodpeckers, especially forest-dependent woodpeckers, are unlikely to cross open water, we know that this divergence here happened after the formation of the Isthmus of Panama. That is, species could not get from South America to North America until that isthmus formed. So that isthmus formed, we think, at this time, because here in clade A we have species that are from North America, including the Cuban ivory-billed woodpecker, and then here in clade B, these are species from South America, and here we have Central America. Okay. So I hope that gives you an idea of how you make a phylogram, so that you can understand what they're talking about when they're talking about biogeography. And we can go over this more in class as well. Okay, so if you go back here, they're telling you that they explain how they do their dating, which I don't think you really need to pay attention to this section here, so I'm just going to delete that. But as you read the text and the results and the discussion, they're going to say that they estimated the average number of substitutions per site, and then turned that into percentages, right? So here they applied the 2% sequence divergence rate that I told you about. So here they're describing essentially what I just told you about. Hopefully the way that I explained it will make this all make a lot of sense. They're going to go into detail about the dates for two of the main splits in that tree. Here they give you those numbers, and then they tell you that they, 1.15 million years ago, based on this software package rates, or it could be 0.91 million years ago if they use a different type of estimate, non-parametric rate smoothing, or if they use Langley-Fitch, or if they use Mega, right? So different methods will give you a different date, and so they're trying to get an idea of the error in that rate by by using multiple methods. So you'll want to figure out from here what's the range of ages for these different divergences. So for instance, if you go back to the figure, this divergence here, where we have the split between up here is ivory-billed woodpecker, and then down here we have Cuban and imperial woodpecker. So this node right here, they're telling you that that could be anywhere from 1.05 million years to 1.57 million years. And then they're also going to give you the dates for this node as well, so you want to write those down. Okay, they're going to give you, they're going to point something out, and this is where it's going to start talking about bootstrapping values. So let's talk about what the nodal support values mean. Carmen gave you a very quick introduction, and I'm going to reinforce that now. So they are telling you that they did bootstrapping here, and that they also used a Bayesian analysis that gave them a value called a posterior probability. But what they are pointing out is they have a statistically a polytomy. So what does that mean? Statistically, a polytomy means if you go in here, and let me clear these out so you can see what I'm talking about. They have 3 different numbers listed for every node. For every split, they have 3 different numbers, okay? The first number here is going to be a bootstrap value. The second number is another bootstrap value, and the third number is a posterior probability. These are all estimates of how true that split really is. So a bootstrap value is an estimate of the amount of error in the tree. So let's take a quick look